Hello and welcome back to lecture 19 of this bio microelectromechanical system. We will begin again by a quick preview of the last lecture. We tried to cover some of the basics of the electrophoresis process as you know we normally do it. It's essentially the motion of a charge in an external electric field through a sieving media. We also talked about certain basic derivations of electrophoretic mobility, velocity, so on and so forth. We demonstrated some gel electrophoresis of DNA in laboratory scale, essentially something where you can do size based fractionation by moving it through a gel and trying to determine or make various mobilities on the basis of length as opposed to frictional forces that a molecule would kind of experience as it moves through a series of sieves in the nanometer scale. We also tried to kind of estimate what does a DNA analysis lab normally comprise of, what kind of components it has. It has a DNA preparation unit, a mixture unit, the PCR thermocycler. It has again electrophoresis mechanism and the readout bench, and also talked about some concepts where this whole lab can be miniaturized onto a single chip level. Okay, we then described or started talking about capillary electrophoresis, which is an offshoot also of the human genome project and the rapidity that was dictated by this project, and we will try to kind of continue in this area and explore this a little more. So capillary electrophoresis essentially is the electrophoresis of DNA in thin microcapillaries 70 to 100 microns in thickness, and one of the advantages that capillary electrophoresis has to offer is in terms of a higher surface area to volume ratio of the gel material which is filled in a channel, and due to this surface area prominence there is a huge heat transfer coefficient, so that the way that heat is transferred to the environment is increased tremendously, which allows us to go for a higher amount of voltage or to sustain higher amount of fields without the gel getting melted. Okay, and therefore this can be utilized to an advantage wherein these high voltages or fields can be used to rapidly translate and fractionate several base pairs of DNA. The rapidity can be to a scale which is almost 1/60th times in terms of times of fractionation from normal gel electrophoresis process. So this right here is an illustration of how it would really look like. So as you see in this figure here, we are talking about a case where there is a crisscross channel, right. This channel here right now is essentially for loading the DNA sample, okay, this channel here, and the other white channel as you are seeing here is essentially for translating the DNA and causing the electrophoresis to happen. The loading can take place either electrokinetically, where the DNA is injected into this gel material here at this particular juncture by giving an electric field, a separate electric field. A perpendicular electric field in this direction is provided to kind of translate whatever has been trapped in here for the DNA and into this particular capillary, right. So if you essentially present a negative electrode okay, so if you present something like a negative electrode here, excuse me, and a positive electrode here, there would be a tendency of these DNA molecules to move through this gel, okay, and as they move they would kind of fractionate, and the sizes as is obvious here in this channel would be separated, okay, and you can do a readout by using either a capillary a waveguide system or something which can be integrated onto the same planar level. This is a black and white mask. This is another mask of a different dimension, and these are some of the images of how the final device looks like. So essentially it is a glass PDMS device. We have talked about replication processes using PDMS earlier. A mask is prepared, a master is prepared, and the device is made in two layers with a lower glass, transparent glass layer bonded to an upper PDMS layer which has the channels carved in or molded in or replicated inside, and there are ports essentially for loading unloading the gel material as well as the samples. And this is a scheme again where it shows how the samples are loaded. Here as you see, there is a green plug like flow here of the DNA sample. There is a thin plug which has been inserted by continuous flow in this empty channel through this cross channel, the loading channel, and once this plug is kind of formulated here, there is liquid gel which is poured from both sides, and this is adjusted accordingly. The idea is that this gel, when it gets into the capillary, gets almost immediately solidified. So therefore you have to be very careful about the rapidity with which it needs to be done. So there is the gel from this side and then another gel solution from this side. When you allow it to cool, it develops a thin gel layer inside the capillary with a plug, the green plug here containing the DNA. Now if you send a positive and negative electrodes through these essentially reservoirs, there is a tendency of this DNA to move towards the positive, and as it moves it kind of fractionates and tries to light up. So the way you make these devices using fabrication strategies for microfabricated PDMS glass capillaries in the following manner: you take a mask here and essentially you pre-clean a glass slide and then spin coat some photoresist material and expose selectively using this black and white transparency mask, so that you can actually create these features on the photoresist material. Okay, this is a negative tone photoresist, maybe SU8 obtained from a company called Microchem. You then follow it by actually pasting these inlet outlet ports over this particular master which is formulated, and then essentially close or place this master inside a a kind of petri dish and pour PDMS on the top of this master. Before doing that, you have to do some surface pre-treatment here, where you can actually make the surface highly hydrophobic so that it can be able to not do away with sticking without sticking to the PDMS when it hardens from the liquid state. Okay, and then once this is done, you can remove the ports along with the PDMS with the channel like structures, and then plasma paste or plasma bond it over this lower glass slide, plain glass slide. Okay, and that would give you a device something looking like this. So you have these small ports here on all four sides, and this essentially is the crisscross channel that you can see. These are real-time optical micrographs of such devices. So these are some detailed results, and this has now been published as well. So as you see here, the following observations are made. If you apply a 300 volts across these two electrodes or these two reservoirs with the DNA somewhere here, and with a voltage of 300 volts, after 25 seconds you see that there is a plug like flow of the DNA. Okay, so this really is and this is the ladder that you are translating. Okay, so this is essentially a 100 to 1000 base pair DNA ladder. So when you are flowing it for about 25 seconds, you see that there is a small stain that is developed and lit up, and this whole assembly is essentially placed over a transilluminator. Okay, this is placed over a transilluminator, and that's why you can see or visualize this particular stain of interest here. You use a 1.5% agarose solution in this microchannels. Now if you keep applying this for another 50 seconds, another 25 seconds, it kind of splits up into these various stains or bands in the same capillary, as you can see. So this is one of the finest examples of capillary electrophoresis. Okay, and Essentially the mobility can be calculated in such stains as this 9.1 10 to the power minus 4 centimeter square plus per volt second. The velocity of the stain would be about roughly 0.078 centimeter per second, and the electric field that is used is almost close to 85.7 volts per second. If you compare time wise to the conventional electrophoresis, how much time reduction happens in this case. In the conventional electrophoresis normally occurs in about 35 minutes or so. Okay, this is a conventional electrophoresis where the ladder is split up in about 35 minutes as opposed to only 50 seconds here. So there is almost close to 1/30th or 1/40th time reduction if you shift from the conventional to the capillary. Okay, so it's basically a reduction of 40, a factor of 40. So it's a huge advantage to the industry because we essentially look for rapid ways and means of doing this DNA fractionation. Okay, and this is one of the best, the very best methods which are available in a capillary where you can do the same job. Okay, so this is another illustration where you can show electrophoresis happening inside a capillary using a 750 base pair porcine reproductive and respiratory virus syndrome. It's essentially a dreaded disease in the swine herds in the United States and across the other part of the world. So essentially this virus is recognized. So it's a viral disease, and the virus is recognized by the 750 base pairs of a certain section of the genome of the virus isolate. Okay, so using agarose capillary and about 300 volts, we have been able to successfully demonstrate electrophoresis in terms of these stains, as you can see through in the capillary. So the mobility is calculated here at about 7.84 10 to the power minus 4, velocity of the stain being about 0.0784 centimeter per second in a field of 100 volts per centimeter. And in this second trial here, the mobility slightly changes to 7.59 10 to the power minus 4 centimeter square per volt second, and the velocity of the stain is 0.065 centimeter per second, and electric field about 85.7 volts per centimeter. These are some of the facts and figures which are important for knowing about this electrophoresis process a little bit more. Another fantastic example is this from this paper published way back in the early 2000s by essentially the Matisse group up at UC Berkeley. So essentially here what Matisse and his group is trying to show is a set of capillary array. Okay, this is instead of one capillary, many capillaries on a radial plate like a CD, and there is a principle which is involved in the detection of DNA stains in this particular illustration. So if you really look at one of the channels here, let's say we want to have a look at this one channel. Okay, essentially something like this. So you have two channels in this one structure or one unit of the channel that is mentioned here, independent channels, two independent channels converging into this port here, which is also at the center and has a common anode. Okay, so it is a positively charged plate which is also a dispensing reservoir. These two are the detection arms on both sides, and these arms are adjusted in a manner that you have the sample ports outside, and you have a crisscross channel on both sample ports from both sides of these main detection arms, and these are all connected integrally to something like a waste collection center. And then you have a cathode which is placed at this other end here of the microchannel, and an anode which is placed somewhere here in the waste reservoir. And then the idea is that when you load the sample here and make it negative, the sample negatively charged, by putting another electrode, the sample kind of electrokinetically gets injected into this detection arm and this detection arm, and then the cathode and anode combination of these two across these two ends are able to, these two ends, this and this end, they are able to drive the small samples which are caught in this detection arm all the way through the arm, and in the process there are stains which are formulated in these two regions here. And essentially the CD has an additional advantage that you can actually calibrate and place a reader detector system which can actually go back and forth radially and scan what all stains are there on this capillary. So there is a unique combination of the rotary motion of this particular microcapillary containing CD and in relation to the radial motion of the detector sensor assembly, and that can help you to kind of identify at least 96, these are all 96 in number, okay. The separate channels are 96 separate channels are there in the CD like platform. So you can actually at a time read about 96 particular reactions, PCR reactions, using this particular capillary. And so this is also a very high throughput process that Mattis and his group has developed before. You can actually also take this particular channel and introduce a lot more turns and accommodate a lot more length of this channel. The only thing here is to be a little careful in designing the way that these channels will twist and turn, and that should not be done at the cost of the resolution loss of the DNA. So there are some interesting work which has been done in this capillary array electrophoresis area by various researchers around the world, okay. So after doing this electrophoresis basics, I would like to turn your attention towards another very important aspect known as the design of a spatial or a space domain PCR reactor. So when we were talking about the PCR reactor in general, we assumed that there is a small chamber contained in time, in space, and the temperature contained at a fixed spatial location and contains a small volume of this PCR fluid, and we assume that the thermal cycling is done on this fixed spatial chamber through on a temporal basis, so with a time varying basis. So these devices are also known as the time domain devices for PCR microreactors. However, one issue with these devices is the very fact that you have to kind of heat a large amount of thermal mass associated with the wafer or the substrate or the base which would contain this chamber in it, and therefore every time you have to ramp up the whole mass to a certain temperature and ramp it down in order to be able to quickly do thermal cycling. However, this is a very inconvenient module, because the thermal mass and particularly the materials that we use in microfabrication, silicon, glass, then your polymers, they are all poor thermal insulators, and therefore it does not give a very easy solution in terms of a rapid ramp up or ramp down. So another approach that was very intelligently thought by a group up at Oxford, Andreas Mann's group, way back in about the early part of this particular decade, was about space domain PCR reactors. So instead of one location and heating at various temperatures of that location, what this group for the first time thought is that why not have 3 different heating zones on the same chip, one heated to 95, one heated to 72, another heated to 50, and then you essentially move the PCR fluid around in these 3 different zones by a serpentine path, okay, and in a serial manner, so that the cycle, which is essentially a 95 followed by a 50 followed by a 72, is followed, and at the same time the PCR gets executed only by virtue of the motion of the droplet in these differentially heated areas of a single surface, okay. So this is called this is a very novel approach, okay, and also called space domain PCR devices. So I would like to draw your attention to how such a device can be designed. It's essentially a challenging engineering problem. So you have 3 different heating zones maintained at different temperatures, and you have a serpentine path over these zones in which you are flowing or circulating the fluid. So when it goes to the first zone, you have to give it time enough for the volume to go up to the temperature of the surface, that is, let's say 90 degrees, and then also you have to wait long enough for the whole denaturation step to happen. So the total amount of time that this small droplet should be present in the 90 degrees heated area is about the ramp up time of the mass of the droplet to go to 90 degrees plus the denaturation time. Similarly, the amount of time that is needed to hold the drop in the 50 degrees area is essentially the ramp down time to the 50 degrees area times of the amount of time it would take for the sample to be present in the extension step, okay, or the annealing step actually. Similarly goes true for the extension step. So you have to design effectively the velocity of this droplet in a manner so that length, which is also equal to velocity into time, would be able to accommodate or give sufficient time for the denaturation, the annealing and the extension process of a PCR to occur in an easier manner. So let us do an example here. We now start designing such a spatial PCR reactor. As you see here, there are different zones 95, 75 and 56 designed in a manner so that you can use probably microfabrication techniques to design this kind of a thing. So you have the 75 degrees only en route between the 56 degrees and the 95 degrees, okay. There is an input output reservoir here, and the understanding is that the droplet would be moved in a direction so that it is able to rest on this particular area, whatever the temperature is, for a time duration where the whole effect can be felt in terms of a successful annealing, a successful extension and a successful denaturation time, okay. So the assumptions that we have to make for solving this particular question is that the denaturation, extension and annealing time are in the ratio of 0.5 to 5 is to 0.5 seconds respectively, and we also know that the spatial reactor has flow channel etched in glass with a channel depth of about 40 microns. The width is around 90 micrometers. We also have flow rates ranging from 6 to 72 nanoliters per second. That is the kind of flexibility that we have, and we have to design the dimensions of this differential temperature zones, okay. The length dimensions or the width dimensions of these differential temperature zones. If we assume that all the zones have the same width, okay, so we are assuming that the zones, the three temperature zones here have the same width. So we have to first take into consideration some properties. So we assume that the sample has properties of water. All PCR fluids are basically aqueous based. Density would be about 1000 kg per meter cube. Specific heat capacity per mass of water is about 4182 joules per kg Kelvin. Thermal conductivity is about 0.06 watts per Kelvin meter. So the thermal capacitance, if you look at, of this particular channel, assuming that the length segment is about L, okay. So you have L as the length segment of the particular channel. So, C thermal will be given by be given by m times of C, which is again mass is essentially density of the medium times area times the volume. Now, you have to see what the area in this case is. So, the width of the channel being W, and let us assume L to be the total length of the segment. The WL is the facing area. So, this is the heat face, okay, of the channel, and of course, the heat has to travel through the thickness of the channel D. So, WLD essentially is what the volume of the channel would also be, okay. Now, if you look at the thermal resistance in this case, the thermal resistance from bottom of the channel to top of the channel is estimated as R thermal is the depth D by K times of square of L, in this case square of the A or square of essentially the area. It's the area. So, area in this particular case is also W times of L, and therefore, the time constant for this thermal circuit, which is also equal to R thermal into C thermal, is essentially in this case rho C D square by K, okay, and this comes out to be, assuming these various values of density, specific heat per unit mass C, the D value of about 40 microns, and also the thermal conductivity of 0.06 watts per Kelvin meter. This comes out to be 0.011 seconds, okay. That is how the whole time constant of this thermal circuit would be denoted by. So, for designing an MEMS based system, what is important is also its reliability, okay. And here the question is that there is a small droplet which you are moving through different zones of heating on a chip, and you have to be 100% sure that the droplet gets the time that it needs for executing the whole denaturation time, along with the time that it would need for going to that temperature, which is 3 times of its time constant. And assuming that you have a maximum velocity of flow, you can give the maximum tolerance to the system, because anything lower than that would be definitely able to get that time of heating and the time that the whole denaturation would take normally otherwise to get completed. So, therefore, we will do all the designing based on the maximum flow rate, which is about 72 nanoliters per second, as given in the problem statement, was the maximum flow rate, okay. So, let us first find out what the linear velocity u would be in case the velocity of flow or the volume rate of flow is maximum. So, linear velocity is actually the volume divided by the area of cross section, which is W into D in this particular case. D is about 90 microns and W is about, D is about 40 microns and W is about 90 microns. So, it is 72 10 to the power minus 12 divided by 36 10 to the power of minus 10, okay, and this comes out to be equal to about 0.02 meters per second. So the time required for passing through the denaturation zone, assuming the denaturation time to be about 0.5 seconds as has been given in the problem statement again, is essentially the total time that this droplet should be in the 95 degrees zone is 3 times of time constant, just 0.011 seconds times 3 plus 0.5. This comes out to be 0.533 seconds, and therefore since the length of the channel needed in the denaturation zone is only, so therefore the length of the 95 degrees Celsius portion of the channel should really be equal to this time here, 0.533 times of 0.02, which is actually about 11 mm. Okay, and since we were actually talking about a flow in this denaturation zone which is twice the length, the length becomes equal to 5.5 mm. We do assume that this portion here is essentially not very major length in comparison to the length of the zone. This turn is essentially neglected because of its smaller magnitude. So the length of the zone that we have been looking at is really about 5.5 millimeters. Now the same length is true for the extension case, because we do not have the annealing case, because we do not have a different time in the annealing area. But for the extension case, as you see here, the time is about almost 10 times. Okay, so let us look at how much velocity will be able to, or how much length will be able to cover in the extension zone. So the time needed for the extension to happen is essentially 3 times of 0.1101 seconds plus 5 seconds, so 5.033 seconds. At the velocity of 0.02 meters per second, the total length that it would need to stay or cover, or the total channel length that it would need to be in this extension zone is 0.02 times 5.033, which is essentially what the total extension length would be. So this is about 100 mm. Okay, assuming this was in meters per second, this is about 100 mm. So again, the very fact that you have one turn which is corresponding to about 0.55 or 5.5 mm, it's a design constraint that you cannot go over this in all the 3 temperature zones. So you have to actually ask the channel to serpentinely turn in this extension area, as can be seen here in the figure. Okay, so this area essentially you have to ask the channel to turn multiple times in this particular area so that it can get heated as it goes into the serpentine path. And so the number of turns that this channel would need to execute in the 72 degrees area is 100 divided by 2 into 5.5, which is essentially about 9 turns. Okay, so you have to design the PCR in a manner so that you have a space here which comprises of about 9 turns of this particular channel, where the space is heated to about 72 degrees Celsius. So the annealing zone characteristics are the same as the denaturation zone, although in the extension zone you need to serpentinely coil the channels about 9 times in order to get the total time of extension, so that the whole DNA pair can be copied. So this in a nutshell is how you design a space domain PCR device or a PCR microreactor. Okay. So I would now like to actually delve into another very interesting area of biosensing, and that is essentially what you can do using the self-assembly skills of a DNA molecule, a deoxyribonucleic acid molecule. So this is so what we are going to do now is to kind of try and discuss some of the various detection diagnostics protocols that has been promulgated by the complementarity itself in the DNA chain and its ability to get self-assembled over one another. So one of the first papers in this area is that generated by Alvisatos and his group. Essentially is about gold nanoclusters. So what he found out is a DNA mediated assembly process way back in 1996. Okay, so here as you see there are 2 complementary strands of DNA which are exactly complementary when they bind to each other with high stringency, and what he has essentially done is that he puts these gold nanoparticles on both these strands separately and let them self assemble, so that there can be an array or an arrangement of these gold nanoparticles. So the moment that these 2 strands are totally complementary to each other, and in fact this is not the only binding kinetics that one may have here. One may have something like, let's say you have this DNA molecule with the gold nanoparticle here, and you have maybe another molecule with a kind of complementary structure which is corresponding to few base pairs here, and then it has its own base pairs which again bind to a third molecule. Okay. So it binds to another third molecule here which has another of these particles, and so therefore there are several such strands which by means of complementarity between the strands would keep binding to each other. Okay. So that is how these self-assembly, the DNA mediated self-assembly process would happen. All right. How you get to know whether the DNA are self-assembling is by looking at what happens after giving sufficient time for these particles to assemble. So this is essentially a scanning electron micrograph of these different gold particles. As you can see, they form these clusters. So these are gold nanoclusters, which is indicative of the fact that they are bound together or held together now by the DNA functionalization which these gold nanoparticles have individually on their surface. Okay. So essentially this is one of the first experiments which demonstrated that DNA mediated self-assembly, how this approach can be used for putting together nanoparticles. So essentially a DNA strand is specific to its complement. So use DNA as an address level, an attachment system to assemble objects, and the DNA can also be attached to these gold particles by using thiol chemistry. So you modify one end of the DNA through a thiol SH group and essentially adsorb the SH onto the gold nanoparticle, which just by pure adsorption it can kind of conjugate, or it can adsorb and then become integral with one of these side chains of the DNA. So SH forms a metal thiolate bond, which is also the basis of the attachment here. Another very interesting work which has been probably reported by the same group is one of the fundamentals of genomic detection. This is one of the first few works which indicate the concept of DNA hybridization. Here, of course, what happens is that you have these avidin coated polystyrene beads. As you can see, these are the avidin moieties on the top of the polystyrene beads. Okay, and avidin is essentially again a vitamin which bonds very well to biotin, which is again some kind of a protein. So essentially what you do here is you coat avidin on these polystyrene beads, and you make a series of capture probes with some particulates at the end, which could actually be immobilized on the top of maybe a gold substrate. Okay, so essentially you are immobilizing this half stranded DNA molecule, which is also a capture probe, onto this gold surface using some thiolated chemistries. Now what you do is you bind the target with the biotin. So you essentially biotinylate the target. There are ways and means of doing it, and I'm not going to get into the chemistry details of that, but essentially there are kits now available through which you can biotinylate proteins, DNA, all sort of biological entities can be biotinylated, which means that a biotin group can be added onto the entities externally. And essentially once this biotin target has been realized, this when you flow the target, and it's going to find its capture probe and getting bound to the capture probe, okay, at the location that it's intended for, and the biotin moiety on the capture probe are able to trap the avidin which you have already coated on the surface of this polystyrene bead. So therefore whenever there is a target binding to a certain capture probe, it is indicated by a polystyrene big bead with the biotin surface function with the avidin surface functionalization in turn binding to this half or this semi DNA or this small part of the DNA tucking out of the hybridization assay. This bonding here between the yellow and red is also known as DNA hybridization. Okay, so how do you detect such a mechanism? So you use an AFM for doing that. Here if you look at the two scans, in the top scan essentially there is a control sample with no capture, so there are no beads which are immobilized, polystyrene beads which are immobilized, as opposed to the captured gold nanoparticles. You can see here that these polystyrene beads have been immobilized onto the surface by virtue of the avidin coated to this target DNA, or sorry, the biotin conjugated onto the target DNA, bound to the immobilized capture probe on the surface. So by virtue of this, it's like a hook which holds the polystyrene bead in place, and therefore you can easily see that the presence or absence of a right capture probe corresponding to the target would just make a difference in terms of laying out a signal as a bound polystyrene bead over the surface. Okay, so this is another very interesting example of how you can make hybridization arrays between a target and a capture probe DNA molecules. These are some other illustrations where the exact same thing has been done, but here as you see there are about 8 microns gold dots over which the capture probes have been immobilized by using thiolated chemistry. Okay, so essentially these dots here that you can see are 8 micron gold dots, okay, made of gold. And what we do here is that in some of the dots we do not bind any capture probe, and in others we do bind capture probes. So essentially you are making some control and some reference sites of binding. So, wherever there are these dots which kind of have the presence of the capture probe, there is always a capture of the polystyrene beads, as can be seen here in the close up view. These small moieties here are the little polystyrene beads, the tiny beads which have gotten captured because of the presence of the capture probe. Okay, so optically you could actually using an optical micrograph or an optical microscope realize whether there is a binding going on between the target DNA and the capture probe just by looking at how these gold dots are, whether they are with polystyrene bead or not. So the controls for some of these experiments are non-thiolated attachments with hybridization, thiolated attachment with non-complementary hybridization, and essentially all PS beads, all polystyrene beads are avidin coated. So then there is no bead capture, indicative of there is a control. So if there is a thiolated attachment, however, and a complementary hybridization with biotin, avidin coated polystyrene beads capture on the patterned gold surface, which gives us an idea of what is the particular target, because if it is complementary only then it is going to bind, and we do have information on the capture probe which is going to bind it. So we can just figure out what is the complementary of the capture probe, which is essentially the target in this particular case. So now we come to another very interesting area, which is DNA hybridization microarrays. Now this area is essentially a very big business nowadays. Billions of dollars are involved in this diagnostics. One area which is about building of DNA microarrays. Okay, these arrays can be used for a variety of applications, including hybridization arrays and for different applications like this RNA expression monitoring applications, sequencing, HIV resequencing, in general trying to detect target DNA or sense some target DNA, so on and so forth. And the very interesting aspect of all this is how do you build such microarrays, or what is the basic principle or a basic mechanism of such microarrays. So definitionally, hybridization essentially is the basis of detection of the unknown nucleotides. Okay, examples that come into picture are biochips for identification for DNA, hybridization of an unknown fluorescently tagged strand with many known strands. Reaction will determine the sequence of the unknown or vice versa. Okay, so if you have an unknown target and you are wanting to hybridize it with something which is known, some capture probe which is a known sequence, and you are able to somehow fluorescently tag the strand with many known strands, okay, the reaction will determine the sequence of the unknown or vice versa. So strands can be lithographically or electronically defined at a specific location. So there are two companies in the world which principally makes these microarrays. One of them is Affymetrix, which does a molecular buildup for the capture probes, using light directed synthesis, and the other company which builds these are Nanogen, and they kind of give this more to the user, and they just build the electronics, wherein electronically you can direct a particular capture probe to a certain site of interest within the micro scale architecture that they would provide. So let us look at what these microarrays are. So essentially it is all about building different sequences or a library of different sequences of DNA onto a single chip level. Here in the left figure, essentially if you are looking at these numbers here, S1, S2, S3, S4, S5 all the way up to S9, these are different locations on the same chip, and if we can somehow direct a specific sequence of a capture probe of single stranded DNA onto this area S1, or the first column and first row, or the second column and first row, and we have an accurate information of what is what. That means what sequence is there in S1, what sequence is there in S2, so on and so forth, up to what sequence is there in S9. Then if we bind the DNA with a fluorescent label, which is the target DNA essentially, onto this and wash this plate, only the bound will stay back, because you have already immobilized the capture probe on this plate, and the DNA which is fluorescently labeled is getting immobilized to the already immobilized captured probe. So it is essentially a hook between the plate and the new target molecule, and that way gets bound. So in case if you wash this plate, the binding goes away, or the binding stays back, or the bound DNA stays back, and you still have the fluorescent signal, which is indicative that there is binding or there is complementarity in the input strand, the target strand, based on which you could find out what is the sequence, because you already know what is the sequence on the capture probe. So this essentially is what the DNA hybridization principle is. And the microarray, the name as the name is indicative, is essentially it's an array of these different sites for doing or immobilizing different capture probes onto this particular site. Okay, so basically that kind of brings us to the end of this lecture. What is interesting here is that we would now be in probably the next lecture working on some of the ways and means of placing these capture probes on the arrays, or building these capture probes on the arrays, and we'll be looking at some of the companies like Nanogen and Affymetrix and the way they immobilize these capture probes and build microarrays, and so essentially that will be covered in the next lecture. Thank you.